So to continue with the previous all whatever classes we have done, we already know about the structure of the nephron, the functional unit of the kidney, and we are very much familiar with the way like the process of formation of urine takes place. But right there during the process of formation of like urine, there will be one particularly an important mechanism that actually occur in this particular place that we should discuss. This mechanism is known as the countercurrent mechanism. But before going on to the countercurrent mechanism, let's learn about the name of the nephron first and foremost. See, as you have already seen that the nephrons are arranged in the medullary region, but in reality the nephron is not necessarily arranged in the medullary region. There are two forms of nephron that will be found. One which is in the cortex and the medulla, entire both the places, and one which is majorly in the cortical region only. So glomerulus will be always found in the cortex region. It will never be found inside the medullary space. So it will be found in the cortex area. So right there always we will find that the glomerulus are arranged. It is the Bowman's capsule which will capture it around that particular space. So Bowman's capsule will be found, or the renal capsule will be found always forming near the in the cortex region, in the cortical region of the kidney. Now right there, after that the all the renal tubule will emerge, and the renal tubule will be arranged in this two different fashion. You can check out easily what are the differences there. In this case, the proximal convoluted tubule is there. It will extend into the medulla because this portion is the medulla. So it will extend into the medulla. Then you will see that the long Henle's loop is formed, and then it will go out and then turn out to be the distal convoluted tubule, and finally it will merge into the collecting duct. But in this case, you find that there is a small proximal convoluted tubule and a very tiny Henle's loop with the proximal, I mean, so distal convoluted tubule right there. So this is like these two different forms of nephron that will be found. This portion, this type of nephron is what is known as the corticular nephron, and I guess you can see the name there, corticular nephron. This type of nephron, they are known as that. Okay, and this type of nephron is known as juxta medullary nephron, or sometimes they are also called as corticomedullary nephron. Both the names are there. Now in this case definitely there will be a lot of use of the Henle's loop section that will be found, but in this case too much of exploitation of the Henle's loop will not be seen. So majorly what is the purpose of this Henle's loop? If you remember from the previous class, it was discussed there that in this particular region that we find that the water is getting reabsorbed back into the blood vessels. The vasa recta that is running adjacent to it, into that vasa recta you will find that the water is moving out. Now for that particular process, the consistency of the blood is maintained, and also the formation of the urine takes place properly. If you remember, it was discussed that the glomerular filtration rate for a day is 180 liters, means a day the kidney process out and form 180 liters, or rather we should say this process out 180 liters of blood and forms the urine, but the final urine is not 180 liter but only 1.5 liter. So there is a tremendous reduction in the urine formation, or rather we must say there is a tremendous concentration of the urine takes place, and that concentration it occurs majorly in this particular section. So we can say rather than this one, this type of the nephron is far more essential for formation or concentration of the urine. So right there the process of counter current mechanism or counter current multiplication is what we can observe. Now let's talk about the structure. So you will find that the proximal convoluted tubule will turn into this tiny thin pipe known as the Henle's loop. But the Henle's loop, when it is moving back up and forming this, will become this later on this distal convoluted tubule. During that time, it will actually widen up slightly and it will become a bit thick walled, because Henle's loops are majorly made up of thin walled like tissues, but here right there it will actually become quite thicker. So this section is known as the descending loop of the Henle, and this section is known as the ascending loop of Henle. Need to remember these two parts, descending loop and the ascending loop, because the flow of urine through this is actually downward and through this it is moving upward. And right there you can notice that there is actually counter flow of the urine is taking place, because urine is getting collected right here, it is traveling through the proximal convoluted tubule and then moving down through this Henle's loop, descending limb, and then it is moving up through the ascending limb, and then it is going to the distal and to the collecting duct. But while doing that, we can clearly see there is actually a counter current or a counter flow of the material is taking place. So this counter current, it actually what helps in that concentration process. Well, this is not alone, there are far other factors that are responsible. First of all, whenever we talk about concentration or anything like that, solute, solvent material, something like that, the point that we should mention here is osmolarity. Osmolarity is a way of understanding the concentration of the fluid or the system that we are talking about. So let's consider this is a system. So in this particular system, we will find the osmolarity of this section and the osmolarity of this section, it varies a lot. How much? The osmolarity value of this particular place, means the cortical region, it is majorly 300 milliliter, you can say milliliter osmol. Okay, 300 milliliter osmol per liter, you can say. This is actually the unit how it is actually measured. Okay, so if it is 300 milliosmol, it is there. So what about this particular section, this bottom section, which is, you can consider the depth of the medulla. Okay, so depth of the medulla, we will find right there its value is somewhat in the range of 1200 to 1400. I'll simply write osmol here. Okay, so it is 1200 to 1400 osmol range is maintained right there. So if we see the change from 300 to 1200 osmol, that means there is at least 4 times more osmolarity can be noticed here. Means right there a tremendous increase or concentration of material is taking place. Now what about this area? Means I'm talking about this area now, the cortex and the medulla, and I'm talking about this particular place, the osmolarity of this particular place. A while ago I just said that the osmolarity, it majorly determine or it actually explain the level of which is something in the yeah, we can say it is something which is in line with the concentration of that particular solution. So that means both these places have some sort of solution there, and it is not incorrect, it is and it is actually true. Both these places actually have interstitial fluid, which actually is like the kind of a fluid which is present between the tissues. So the cortex region also have an interstitial fluid, the medullary region also have an interstitial fluid, but the medullary region of the medullary interstitial is somewhat, it has gradient of its own. So if we find 300 osmol right in this particular place, then it will keep on changing as 400 osmol here, and then we'll find somewhat around 600 osmol in this region, then we'll find somewhat around 900 to be there in this region, and so on and so forth, reaching out towards the 1200 mark. Okay, so it is gradually increasing. Now how this gradual increase is possible? So for this gradual increase, we should remember the thing that is responsible there, or the way it is all being possible, is simply by conservation of energy. Now, how conservation of energy is working there? See, it's simply this section, the way these cells are arranged or the way they are made, the proteins that they contain, it all matters to that. Right there in the descending limb, the wall is like the cuboidal epithelium that it is made up of, they have a tendency of diffusing water out. Means from there we can easily find water can come out very easily, very easily. Water is coming out. Now this water, it will be absorbed by the vasa recta that is flowing adjacent to it. So let's draw the vasa recta also right there. So the vasa recta is right there. Now this vasa recta will simply absorb all this water. Okay, the VR vasa recta. So this is absorbing all this water. So it has a tendency. That's how actually the blood consistency is maintained, isn't it? So this water, it is able to come out of this particular descending limb. Now this process is just because the cells are made such a way it can allow the movement of water, or it is, we can simply say the permeability of water is very high for this particular place. But this area, it is completely impermeable for the ionic exchange, because this fluid inside, it has a large amount of ionic concentration, isn't it? But this ion cannot come out. All this ion will remain inside only. The descending loop will not allow the movement of the ion. Right there in the ascending loop, even if a little amount of water comes out, the major thing that is working there, especially right around this like thicker section of the ascending loop, is we will find that from here, via active transport, okay, via active transport, ions are able to come out, majorly the sodium ions. Okay, the sodium ion, it is able to come out right there from this place. Even this section also, I will not, like this place also the same thing happen. So when this sodium ion, it comes out from this particular place, again I'm saying this is completely mediated by the active transport system. Okay, so active transport means with the expense of ATP, this movement is being allowed, but it will not allow any movement of water out, or even if there is, there will be very tiny movement of water with like very few molecules of water will actually come out. So if that happen, then continuously this like ion that is being sent out, it creates a very high grade of osmolarity in this particular region, because it is increasing the like osmolarity of this particular place, because the more the solute particle, and this will be considered a solute, isn't it? So more of it will create more osmolarity there, and that osmolarity, that change or the gradient in the osmolarity is what allow the movement of water even more from this particular place. And whenever the water comes out, all this water will be taken back into this vasa recta. The vasa recta will simply absorb all this water. That way the entire urine that is coming out from this particular place, it gets highly concentrated in this particular section. Even this portion, the collecting duct also is helping in this particular process, the concentration, how to like concentrate the entire thing. Okay, so even the vasa recta is going through this area. The vasa recta will actually encompass both the side, isn't it? So the vasa recta is also coming from this particular section. So when the vasa recta is in this particular area, the collecting duct has got a transport system for movement of water. So the collecting duct will again send water out from inside to outside. So if the water is coming out from the collecting duct, it is going into the vasa recta again. So then what happens there? Even more concentration of the urine will take place. That's why we find that 180 liters of being urine being processed right there, but only 1.5 liter of the urine finally comes out and gets like micturated from the body. So this is how, this mechanism of countercurrent flow is what is helping in the like formation or concentration of the urine in, or even we can say is major formation of the urine that is taking place in the body. If this thing goes wrong, then the entire process will actually we'll see that the entire thing will actually fall apart. It will actually get very reduced. This countercurrent mechanism is very important for the formation of urine.